molecule responsible for all similarities and differences in people is deoxyribonucleic acid, DNA. That's what determines our heredity. That's what makes everybody different. Okay. It's tightly wound up in structures called chromosomes, okay, in the nucleus of the cell. DNA carries the genetic code to produce thousands of different proteins that make us who we are. So the DNA produces the proteins. The DNA gives you a blueprint that's going to tell what order the amino acids are going to be in to form a certain protein. Of all the 1,000 proteins, we use 20 different amino acids, and that's coded. The order is coded by DNA, deoxyribonucleic acid. The chromosomes are donated from each parent equally. 50% of the chromosomes come from your mom, 50% come from the father. Okay. Each egg and sperm cells have half the chromosomes of the cells they produce. The human body has 46 chromosomes. Okay. If we had 46 chromosomes in the sperm, 46 in the egg, we have 92, and that would not be good. Okay. We have to keep it at 46. Okay. 92 would be fatal. Okay. And 46 chromosome is for humans, not for all other animals. Other animals have varying amounts. Okay. So when we're doing the division of the cells, when we're in the sperm cells and the egg cells, they go through a special process, not mitosis, meiosis, that reduces the number of chromosomes by half. So when they fuse, then we have a total of 46 again. Each one carries 23. Okay. So thousands of genes carry genetic information for thousands of proteins that dictate your form, and some believe our behavior. Some people think behavior is genetic. There have been some studies done that show people with certain DNA sequences that produce certain proteins have more tendency towards violent crimes if they lack that. It hasn't been, it hasn't been verified scientifically yet. That would mean that you could take a person and say, well, you have this gene, so you're going to go to jail because you're going to commit a violent crime. You can't do that. But there are indications that certain genes and certain proteins link people to certain predispositions. And it's interesting research. It really is.
As a breeder,
Huh?
So I used to breed horses when I lived in Colorado.
Right.
And in order to be accredited, like, and I guess it's the same for dogs, you can't breed with a specific trait. So like if you have a male, for example, that's aggressive, he is not good for breeding, because they say like that the aggression towards like humans is a dominant trait.
Correct.
So if you have like a aggressive dog, for example, like let's say she gives a litter and there's 2 males and one of them is super aggressive, you're not allowed to breed with that dog to continue being accredited.
Right. That's the AKC rules are doing. Yeah.
Well, we did horses, but yeah. So right, with the horses too, like if we had a male or that was super aggressive towards other, like the other horses or with us, then we weren't allowed to breed with that male.
Yeah.
He's
So
like considered a stud.
So they are starting to show that behavior is actually part of the genetic code also, which is really interesting research. You know, they used to think it was all strictly environmental, but they are showing that a lot of genetic disposition towards violent behavior, and you can see it in dogs and horses, like you were saying, and in humans the same way. But you can't say, since this person committed a certain crime, you can't breed anymore, like you can with a horse or a dog.
Sure.
We can't do that. At least not at this point. What was the movie? Minority Report. Remember that movie, Tom Cruise?
Yeah, when they take his eye out.
Yeah, he uses his eye for one of the scanners and stuff like that. Yeah, Minority Report is based on that premise. It's based on, we can see the future and we know you're going to commit a crime, so we can arrest you before the crime happens. And it's well done. It's interesting. And this is the same thing. If we can prove a certain gene is going to determine violent behavior, can we arrest that person and get them out of society?
I don't know. I was listening to this crime podcast, and where I'm like 200 episodes in, and almost always they say like if you name your kid Junior, then he's going to become a criminal like you were. But there are some cases though where like the kid gets put up for adoption, and then so he never knows as a biological parents, and his dad goes to prison for XYZ, and then when that kid ends up being an adult, even though he's in a different environment, ends up being coming in prison for like the exact same thing.
Yeah, there's been a lot of studies that. One was called the Brazil Project, and that was the actual cloning of Adolf Hitler to try to mimic those and carry on that gene set. There's a lot of studies that have been done. It's been very interesting. But even if we do now, that's the scientific view. Now, after that, then you got a moral view of once you understand that, what can we actually do with it? Can we actually arrest someone who has not committed a crime because they have the genes that predispose them to behaving violently? So it's an interesting concept. Most things don't surprise me, but some things do. There was an article in the Journal for Neuroanatomy of the brain, and these people wanted to. We can do surgery now when the fetus is in the womb to correct certain things. Like if a baby doesn't have an arm, we can go in there and use stem cells and grow an arm before birth. We can correct hearing. We can correct some blindness issues. We can do certain things now through surgery on the fetus. There was a couple that wanted to. They were suing the Supreme Court, actually, to be able to make their baby disabled. Okay, and at first I thought, you got to be crazy. But then I read the article. They wanted to make their baby deaf. They wanted the baby to be born deaf. Now, at first you think that's absolutely nuts, insane, right? That's your first response. You can't do that. But who's to determine what's right or wrong? These are two deaf parents. Their entire society is deaf. Their friends are deaf. Their group, their whole livelihood is involved in a deaf circle. If the baby's born able to hear, that baby will not fit in to their group. I'm not saying I agree with it, but all of a sudden I see their point. And it becomes more of not a scientific issue, but a moral issue now. And what do you decide?
I think the reason why people don't do it, like don't typically agree with it, is because more cases than not, things are used for a negative connotation in the wrong hands, which tends to be money. You know, like
right.
People get money, and like with the genetic. So there's a 23andMe is like that thing that a lot of people have used to see what percentage of whatever they are, right? So you spit into the cup, and then the company sends a list back to you that's like you're 13% Italian and
right.
Swedish and whatever, right? Well, they also have a 23andMe has developed a database based off of hereditary diseases in your family, and it's a separate site, and they have said that some people are trying to use that to genetically modify their baby for looks.
Yep.
So like instead of being like, hey, this kid is going to have Huntington's disease, let's edit the genes for that. They're like, I want my kid to have blue hair and blonde eyes. And if someone's modifying their kid to not be deaf, I'm sure that people are going to be like, well, what's the difference?
Right. That was the argument. And they made a good argument. I mean, it's strictly determined by society. So it's become very interesting what we can do with all the new scientific advances. The old adage is just because we can do it, should we do it? You know, it's been actually amazing. But yeah, like molecular genetics, and we can detect certain genetic diseases. Right? So it estimated that 3 to 5% of the human population suffers from a serious genetic defect. That's a lot. That's a lot of people. 5% of our population. Right? That's 200 million people. What if genetic diseases could be detected and cured? That's where we're heading. Okay. 2 new technologies, gene therapy and pre-implantation diagnosis, are going to help us to do that. Again, just because we can, should we? That's going to be the issue that's going to come up. For a couple with a history of genetic disease in the family, pregnancy is a time anxiety. Tay-Sachs disease. Okay, Gaucher's disease, lipid storage diseases. These people that have recessive genetic diseases have a 25% chance of having a kid if both parents are carriers of having a kid with the disease. And these diseases like Tay-Sachs disease is fatal within the first 2-3 years. Okay, so if we can change these odds, we can modify that. We can have those odds a lot better for survival rates for these children. So through genetic counseling, these couples can learn the probability that the child has a disease. Okay, so for about 200 genetic diseases, uncertainty can be eliminated. You can tell what the odds are that they will have a certain disease on their baby. Amniocentesis. Okay, that's when we take about 20 milliliters of fluid from the sac that surrounds the fetus. We remove that. And the chronic villus sampling, we remove cells from the fetal membrane. And the two procedures used to obtain fetal cells for genetic testing. So we can test the fetus, what type of cells they have, and determine the genetics, and determine if it has Tay-Sachs, if it has Gaucher's disease. Okay? So we can do it's an amniocentesis and a chorionic villus sampling. So we sample the fluid surrounding the baby, the fetus, and then we remove cells from that cell membrane that surrounds the fetus. So fetal cells are cultured and tested by enzyme assay. We talked about enzyme assays. We use different enzymes to lock onto different proteins that are present, and we can tell if those certain enzymes are there. Okay, DNA test to look for these genetic diseases. If a genetic disease is diagnosed, then the parents make a difficult decision. They can either abort the fetus or carry it to term and deal with the effects of genetic disease. But either way, the parents know that the child has that genetic defect, has that genetic disease. Okay? And then you got a decision, like I said, that's between the parents, make up whether abort the baby or carry it to full term. Scientists have developed a more impressive way to test for genetic diseases before the embryo implants into the uterine lining. Okay? So this technique is called pre-implantation diagnosis. Remember your egg, if you had anatomy, you know the egg and the sperm are fertilized in the fallopian tube. Okay? Then once that fertilization occurs, starts to develop, then it goes down the tube and into the uterus. Once it's the uterus, then it implants. Okay? This technique can tell you if there's disease before the egg even implants into the uterus. Okay? It's called pre-implant. It's a pre-implantation diagnosis. It involves fertilizing a human egg, allowing the resulting zygote as a fertilized egg, sperm and egg come together, to divide into a sterile petri dish. Okay? When the zygote consists of 8 to 16 cells, one of these cells is removed for genetic testing. Remember, it goes from 1 cell to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32, because every cell divides, times 2, and doubles. And that's how cell growth occurs. Okay? Only genetically normal embryos are implanted in the mother this way. So if it doesn't have the correct genetics, it doesn't get implanted. So there's no implantation, there's no development at all. Okay? So the genetic disease that we can detect could be eliminated by pre-implantation diagnosis. Then when you get implanted in the uterus, because only a zygote with what's called good genes is used. Okay, so it only allows the fertilized egg that has no genetic diseases that tested for to implant into the uterus with this pre-implantation. Gene therapy is a second way in which genetic diseases may one day be eliminated. Okay. Foreign genes, including growth hormone, have been introduced into a fertilized mouse egg, and the zygotes implanted into female mice. So we can actually put genes into species that are lacking it now. Okay. Baby mice are born with foreign growth hormone genes who are about 3 times larger than their normal litter mates. Okay. So we induced the growth hormone. We did this at Long Beach VA Hospital. We did it with rabbits. Okay. We did this with Australian rabbits. They did this growth hormone that was from bacteria. We introduced them into the developing fetus of the rabbits. And these rabbits were at least 3 to 4 times larger than a regular rabbit. Rabbits, say 15 pounds, 12 pounds, 3 times. You got 30, 40-pound rabbits now. And these were it was really impressive. It was really amazing what they could do. And this was years ago. This is when I was at Long Beach doing studies. Okay. So this kind of technology needs to introduce normal genes into human fertilized eggs, carrying defective genes. So if you can tell you have a defective gene, okay, you can take a fertilized egg and introduce a gene to it, replacing the defective one, making it, quote, normal. So we can treat some diseases in the womb, like we said. So we can make people, I don't want to say normal, but normal for our standards. And that's what the whole issue of that court case was that we can make what we think normal could be something that someone else thinks normal is different. And like I said, that becomes a moral issue, not a scientific issue. In the 1950s, genetic information was demonstrated to be nuclein. It was called DNA. That's when they discovered this genetic information was carried through in DNA. Okay. In 1953, James Watson and Francis Crick, Watson-Crick, published a paper describing the structure of DNA. They come up with the double helix molecule. Okay. So the structure of the nucleotide, DNA is built on nucleotides. DNA and RNA, deoxyribonucleic acid and ribonucleic acid, are long polymers. Okay, they're very long polymers. Okay, whose monomer units are nucleotides. So you have nucleotides, and these nucleotides, you have 1, 2, 3, 4, up to thousands. Okay, just long chains of these nucleotides. Just have long chains of amino acids for proteins. We have long chains of nucleotides that make up our DNA and our RNA. Okay, a nucleotide, every nucleotide consists of a nitrogen containing heterocyclic base. So it's a cyclic structure. There's a nitrogen atom inside that cyclical structure. Okay, see, that's the base. You also have, and that you have two types. You have purine and you have pyrimidines. Okay, there's two types of bases. Okay, so you have the nitrogen heterocyclic base. It's either going to be a purine or a pyrimidine. Okay, we'll show the difference of those, how they bond together. And then you have a 5-carbon sugar ring. Okay, that's 2 of the parts, and there's 3 parts total. Okay, that could be ribose in the case of RNA, deoxyribose in DNA. If the sugar is ribose, it forms RNA. If the sugar is deoxyribose, it forms DNA. Okay, and you have at least 1 phosphorus group. You can have more than that, but you have at least 1 phosphorus group. So every nucleotide, which is our building blocks for DNA and RNA, okay, every one consists of 3 parts. A heterocyclic base with a nitrogen atom in it, and those are either purines or pyrimidines. A 5-carbon sugar ring, if it's RNA, if it's deoxyribose, it's DNA, and 1 phosphate group at least. All right, so here's our structure. Okay, this is our phosphate group. There's your phosphate group. This is your sugar. And this is ribose in this case. I'm sorry, this is actually deoxyribose. This is deoxyribose. That's your sugar. And this is your ring structure here. And you can have a cyclical ring structure containing at least 1 nitrogen atom. If there's 2 rings, it's a purine. If there's 1 ring, it's a pyrimidine. That's what we're going to be able to tell in a little bit here. Okay? But every single nucleotide has that basic component: a phosphate, a sugar, and a nitrogen base, cyclical nitrogen base. Could be 1 ring or 2 rings. All right. So the nucleotide structure. So ring structure found in both the base and the sugar. Remember, sugars are ring structures also, and so is the base. So the base rings are numbered as usual. Sugar rings are given the designation of prime. So the ones, the numbering, like 1, 2, 3, 4, 5 number of carbons, okay, are just the same as on the base, with the nitrogen atoms in it. On the sugars, we use the prime designation, just to distinguish between the base and the sugar. It doesn't change anything. We use it as a prime: 1 prime, 2 prime, 3 prime, and so on. We're numbering our carbons. So covalent bonds exist between the sugar. Phosphate group is a phosphoester bond. That's the bond that bonds the sugar and the phosphate group. Okay. Bonds between the base and the sugar is a beta galactosidic linkage, joining the number 1 carbon of the sugar and the nitrogen of the base. So the number 1 carbon, remember, when we do our Hayworth projections. We do our structures that look like this, look like this. Remember we had our OHCH2OH group here, and everything else was hydrogen. Remember, if this OH was up, it was beta. If it was down, it was alpha. This is our number one carbon. That's where the linkage is going to occur. It occurs, and we give it a prime because it's on the sugar now. It doesn't make any difference. It just allows you to distinguish a number one on the sugar from number one on the nitrogen base. That's all it does. So this carbon is carbon number one prime. Okay? And that's this one right here. And that's where we're going to bond our nitrogen atom from the base at the number one carbon position. Okay, so this is our phosphate group. That's one of our parts. And these are our sugar groups. You can have DNA, deoxyribose, or you can have ribose. Okay, deoxy means you're missing an oxygen. Deoxy, without an oxygen. So you're missing this OH group right here in the number two position. That's how you can tell the difference between ribose and deoxyribose. In deoxyribose, it's a 5-ring sugar and the oxygen is missing. In ribose, it's a 5-ring sugar and the oxygen is there. So if you're missing the OH group at that number two position, okay, if you just have hydrogen there, it's deoxy. If you have OH, it's ribose. And that's what we got here. You can see the deoxyribose. You've lost that OH group. In ribose, okay, and it's at the 2' carbon. It's our number 2' carbon. That's where you're missing it, missing the OH group. It just replaces oxygen. So just remember, deoxy means without oxygen at the number 2' carbon. Number 2 carbon, because this is our number one carbon. This is our 1' carbon. And this is where the nitrogen bond is going to occur to our base at the number one carbon. That's where you dehydrate, we get dehydration reaction, we lose OH and H, and we bond that covalent bond between the number one carbon and the nitrogen of the base. So the 5-carbon sugar in RNA is ribose. The sugar in DNA is 2' deoxyribose. Only difference is the two sugars is found at the number 2' carbon. Ribose has a hydroxyl group. Deoxyribose has a hydrogen. It's missing the oxygen. All right, major purine bases. So nitrogenous bases, we said the bases are nitrogenous. They have a nitrogen in at least one, and they're heterocyclical. They're in a cycle, in a circle. Okay? Cyclical compounds with at least one nitrogen atom in the ring structure. Those are our bases. And that's one part of the nucleotide. Purines are double ring structures. So if the base is a purine, it has 2 rings, usually a 5-member ring and a 6-member ring. Those are our purines. It's a 6-member ring fused to a 5-member ring. Okay? Again, those are your purines. Okay? So these are purines. This is adenine, and this is guanine. Okay? Those are your purines for bases because they have 2 ring structures. They have a 6-membered ring here and a 5-member ring. 6-member ring, 5-member ring. So if you see 2 rings, you know they're purines. Okay? Pyrimidines are only going to have 1 ring. So major pyrimidine bases, a pyrimidine consists of a single 6-membered ring. Okay, so there's only 1 ring now for pyrimidines, for your base. Remember, you have a base, a sugar, and a phosphate group. Okay, and there's 3 bases that are pyrimidines. There's cytosine, there's thymine, and there's uracil. Now, the difference is in DNA and RNA. DNA has cysteine and thymine. So cysteine and thymine occur in DNA. And then cysteine and uracil occur in RNA. So thymine is replaced by uracil in RNA. That's the difference. So you have 3 pyrimidine bases, single ring structures. Okay. So here's your nucleotide. This is your phosphate group. This is your sugar. And then you have your base nitrogen base at top. If it's one ring, it's a pyrimidine. If it's two rings, it's a purine. Purines have a 5-member ring and a 6-member ring. Pyrimidines have a 6-membered ring. And this is a ribonucleotide because it's ribose. It's ribose because right here, this OH group is present at the number 2 prime carbon. Okay? So it's a ribose. So it's a ribonucleotide. Okay? The next one, you can see you have a phosphate group. You have your sugar. And you have your base attached. Again, either purine or pyrimidine. This is going to be a deoxy. You see the word right there. Deoxyribonucleic. It's deoxyribose because at the number 2 prime carbon, the OH, the oxygen is missing. Everybody understand the difference? It's ribose. It has an oxygen. If it's deoxyribose, it means the oxygen's been removed without oxygen at the number 2 prime carbon. Okay, so that's going to be a ribonucleotide and a deoxyribonucleotide. All right, so nucleotide is the repeating unit of the DNA or RNA polymer. So just like our proteins, our amino acids were the repeating unit, with DNA and RNA, our nucleotide is the repeating unit. You have one unit, one nucleotide, 2, up to thousands. Okay, that repeating unit forms DNA and RNA. Okay, and this is a repeating unit. This is adenine, okay, because this is the adenine base. Okay, this is a phosphate group. This is the sugar. Okay, this is ribose. This is deoxyribose. I'm sorry, deoxyribose. Okay, so right there, it's a deoxyribose sugar, because we're missing the oxygen at the number 2 prime carbon, which is right here. That's your 2 prime carbon. Again, it's a number 2, but we just use that to distinguish between the bases and numbering. Okay, and we have a hydrogen here. We do not have our OH group. So it's a deoxynucleotide, and that's adenine. And that is a purine. It's a purine because the base has a 5-member ring and a 6-member ring. And the base always attaches to the number 1 prime carbon. And the number 2 prime carbon is where we look for to see if we have an oxygen attached there. The nitrogen base is attached to the beta position. Remember, if this goes up, it was beta. If that link went down, it was alpha. So that's a beta 1 nitrogen linkage. Okay, and this is ribo. This is RNA. It's a ribose sugar because it has the, and deoxyribose is a DNA sugar. So it'll be DNA if the deoxyribose sugar with missing the oxygen. It's a ribose if it has the OH not missing the oxygen. Okay, so the sugar is a phosphorylated at carbon number 5. So the number 1 carbon determines your alpha or beta and attaches to the nitrogen base. The number 2 carbon determines if it's a deoxyribose or a ribose. The number 5 carbon, this is your number 5 carbon right here. This is your 5 prime carbon. The number 5 prime carbon attaches your phosphate group. So you have to know the number 1 carbon, number 2 carbon, number 3 carbon, and they're prime because it's the sugar. So 1 prime is your glycosidic bond that attaches to your nitrogen. It's beta if it goes up, alpha if it goes down. The number 2 carbon, number 2 prime carbon, I should say. If it has an H group attached going down, it is ribose. If it is deoxyribose. Okay? If it has an OH attached, it is ribose because the oxygen is present. Deoxy means the oxygen is not present at the number 2 prime carbon. At the number 5 prime carbon, we have our attachment for our phosphate group. All right, a lot of information there. Okay, so deoxythymidine 5-monophosphate is DTMP, the abbreviation. Well, several abbreviations for it here. Okay, so deoxy, okay, thymidine 5-monophosphate. Deoxy, okay, because we're missing the oxygen here. Okay, thymidine, this is the thymine group right here for your base. 5 prime at the number 5 prime carbon right here, we have monophosphate, one phosphate. That's how you break the name down. It's exactly what it is. So it's deoxythymidine 5 prime monophosphate. And it's abbreviated DTMP. So the figure shows linkages between the nitrogenous base, which is right here. This is your nitrogenous base, thymidine, which is the whole thing right here, and the 5 carbon sugar deoxyribose. Again, it's deoxyribose because it's missing the oxygen. And this is your base a thymine. And this is a pyrimidine because it has one 6-membered ring. And the deoxy and the phosphoryl group is attached to the 5 prime carbon. So you have different labels you can do by the different amount of phosphate groups that are on. We can have, if we use adenine, adenine. This is your adenine, the adenine base. This is a purine, has 2 rings, a 6-member and a 5-member ring, and it is attached to the sugar. And this is ribose because at the number 2 carbon, there is an oxygen there. So that's your ribose. Okay. And then you have phosphate groups attached to it. So this basic structure, the base and the sugar is called adenosine. Okay. We add 1 phosphate. It's called adenosine monophosphate. We add 2 phosphates. Okay. It's called adenosine diphosphate. Okay. We have 3 phosphates. It's called adenosine triphosphate. So mono, di, and tri. Common names. Adenosine triphosphate is ATP. Adenine diphosphate is ADP. Adenine monophosphate is AMP. Okay. Deoxy monophosphate is DAMP. Deoxy adenine diphosphate is DADP. And deoxy adenine triphosphate is DATP. Those are abbreviations that we use. Okay, so we have adenine monophosphate, diphosphate, and triphosphate, and then deoxy, depending on the sugar that we have, monophosphate, diphosphate, and triphosphate. Draw the structures for the nucleotides. Contain the following. We have a ribose group, adenine, and 2 phosphate groups attached. So you can draw ribose sugar. The ribose sugar is one that has the OH group at the number 2 carbon. Adenine is the structure that we're going to draw. We're going to look that up and draw the structure of adenine because there's 5 bases that we have, and we add 2 phosphate groups to it. Okay, so ribose, adenine, 2 phosphoryl groups. So this is ribose. This is your sugar. Okay, and it's got the OH group right there. Okay, so it has the OH group present, so it is ribose. So you can draw your 5-membered ring there, and then you're going to draw adenine, which adenine is a purine, 2-membered ring, and just look that up in the chart there of how to draw that. And you're going to add 2 phosphate groups or 2 phosphoryl groups to it. So it's going to be diphosphate. Okay. Next one is 2-deoxyguanine, 3 phosphates. Okay. Again, 2-deoxyribose. This is your sugar. At the number 2 carbon, it's missing the OH, the oxygen. So it's 2-deoxyribose. Guanine is your purine. 5-member ring and a 6-membered ring. And you want 3 triphosphate. So 3 phosphate groups attached to the number 5 carbon. So when you're identifying these, you need to understand, you can look these bases up. That's easy. This base is always going to attach to the number 1 prime carbon. At the number 2 prime carbon, we're going to determine whether it's deoxy or ribose or deoxyribose. And to the number 5 carbon, 5 prime carbon, that's where we're going to attach our phosphate group, whether it's 1, 2, or 3. That's how to draw them. Draw the structures consisting of the following units. Okay, the first one, 2-deoxyribose thymine, 1 phosphate group. Again, you draw your sugar. They're all drawn the same, except the only difference is going to be right here. Okay. Deoxy, so without oxygen, at the number 2 prime carbon. At the number 1 prime carbon, you connect thymine. Thymine is a purine. Thymine is a pyrimidine, only has 1 6-membered carbon ring. Okay, and they're going to add 1 monophosphate group to the number 5 prime carbon. So you have to know where the number 1 prime carbon is, number 2 prime carbon, number 5 prime carbon to draw these. Okay, ribose, cytosine, 3 phosphate groups. Okay, so we draw ribose, which means we include the OH group at the number 2 carbon. Number 1 carbon, we attach cytosine, which is this. And again, that's a pyrimidine. It has 1 6-membered ring. And the number 5 prime carbon, we attach 3 phosphate groups. Okay. Ribose, uracil, and 1 phosphoryl group. Remember, uracil is the pyrimidine, the base that is used in RNA. Uracil is not found in DNA. Thymine is found in DNA. Instead of thymine in RNA, it is uracil. That's one of the differences. So our sugar is ribose, which means we have an OH group at the number 2 prime carbon. Okay, 2 prime carbon. The number 1 prime carbon has uracil, pyrimidine, nitrogenous base attached to it. 1 6-membered ring. And at the number 5 prime carbon, we have 1 phosphate group, monophosphate. Okay. All right, structure of DNA and RNA. They're going to be different. These nucleotides combined, right, form a chain or polymerize in a series of 3 to 5 3 to 5 phosphodiester bonds. 3 prime to 5 prime is just indication of where the bond is. Okay, this is your 3 prime carbon. Remember your sugar ring. This was number 1. This was number 2. That's number 3. 3 is where the phosphate group attaches to the next link in the series. That's the 3 prime carbon, 3 prime carbon, 3 prime carbon. Okay, and then the phosphate groups, like we said, attach to the number 5 carbon. Okay, so you form a number 3 carbon to the phosphate group, and the phosphate group forms to the 5 carbon. So you're going from 3 phosphate group to 5, and et cetera. That's what forms the link right here. Okay, you're attaching from the number 3 carbon to the phosphate, and the phosphate group is attaching to the number 5 of the next one. So we have a 3-5 end. Okay, this is the 5 prime phosphate on one unit, esterification of the 3 prime OH group on the adjacent unit. That's where your linkage is occurring, just like we said there. So the backbone, this is called your sugar phosphate backbone. Okay, this is all your sugar and phosphate, and the bases stick out. Pyrimidine, adenine, cytosine, and guanine. The bases stick out from it. Okay, and it's going to be in a 3 prime to 5 prime unit or a 5 prime to 3 prime, depending how it's oriented with one another. Okay, so this side ends here at your 3 prime. This side ends here at your 5 prime. So that's the direction of the chain. It was like the terminals, like the N terminal and C terminals that we had with our amino acid chains with our proteins. So this is the 5 prime end. And this is the 3 prime end down here. Can't really see it. I'm going to take that off. Okay. So a 5 prime end and a 3 prime end. Okay. It's where the number 3 carbon ends, or the number 5 carbon has bonded to the phosphate group. So the 3 prime carbon is linked to a 5 prime carbon by a phospho diester bond. Okay. And the bases that you have are cytosine, thymine, and guanine. Again, guanine is a purine, thymine is a pyrimidine, and cytosine is a pyrimidine with the ring structures. So you have the sugar phosphate backbone extending all across here. And from that, these bases stick out from it. That's how DNA functions. You have a sugar phosphate backbone and the bases extend out. And that's what's going to form your bond and your attachments is the bases. And the sugar phosphate backbone runs from a 3 to 5 direction or a 5' to 3' direction, depending on how it's orientated. Okay. So DNA consists of 2 chains of nucleotides. They're coiled around one another. It's what's called a right-handed double helix. So think of a ladder, 2 strands, and these bases are attached to each other, the adenine, guanine, cytosine, thymine, and they form the rings of the ladder. And you take that ladder and you twist it. You just kind of rotate it to the right-hand side, and it makes this little spiral. That's a double helix. That's what it looks like. So you have a sugar phosphate backbone. Like we said, 2 strands are spidered on the outside of the helix, like the handrails on a spiral staircase. Think of a spiral staircase going up. That's what a double helix looks like. The nitrogenous bases extend into the center at right angles to the backbone. Okay? As if they're like the steps of the spiral staircase. So the sugar phosphate backbone make up the rails, and the steps are the bases, the adenine, thymine, guanine, cytosine that extend out into it. Hydrogen bonding of the DNA helix. A non-covalent attraction aiding to maintain the double helix is hydrogen bonding. Where hydrogen bonding is going to occur between our nitrogenous base pairs. That's going to hold us, the steps of the ladder together. Okay. Adenine, okay, bonds with thymine. Cytosine bonds with guanine. Adenine and thymine form 2 hydrogen bonds. Okay. Cytosine and guanine form 3 hydrogen bonds. That's missing a bond right here. There, now it's there. Okay. So you got 2 bonds between adenine and thymine, 2 hydrogen bonds, and you got 3 bonds between cysteine and guanine. So A and T always go together, C and G always go together. Okay. This is what you call base pairing. The diameter of this double helix is about 2 nanometers. Nanometers 10 to the minus 9. So very small diameter. The distance dictated by the dimension of the purine pyrimidine base pairs. Okay. So base pairing explains the observation that the amount of adenine always equals the amount of thymine, because adenine always pairs with thymine, and guanine always pairs with cytosine. So the amount of adenine is always the equal amount of thymine, and the amount of guanine is always the amount of cytosine for any DNA sample, because that's what they pair with. It can be a one-to-one relationship. Okay. So complementary strands. 2 DNA strands are complementary strands, which means the sequence of one automatically determines the sequence of the other. If the sequence here, this is your sugar phosphate background. Phosphate, sugar, phosphate, sugar, phosphate, sugar. Remember that linkage? That's your backbone. That's your backbone. So adenine always pairs with thymine. Thymine pairs with adenine. Guanine pairs with cytosine. Cytosine pairs with guanine. Guanine pairs with cytosine. Adenine pairs with thymine. Okay? If you have one strand, you know the other. Okay? Because you know what it pairs with. Okay? So you'll always know the strand. Now the strands run opposite of each other. What's called anti-parallel. Anti-parallel simply means this is the 5' end, so it ends on the 5 carbon sugar, and this is the 3' end. It ends on the number 3 carbon in the 5 carbon sugar. And the strand opposing it or complementary to it is going to run from 5 to 3. They're going to have different ends on each one as they match up. That's called anti-parallel. Okay? So only when the 2 strands are anti-parallel can the base pairs form the hydrogen bonds that hold them together. So when the strands have to line up to form those bonds in between there, between adenine and thymine and cytosine and guanine, to hold this DNA together, they have to be anti-parallel. One goes 5 to 3, the other goes 3 to 5. Okay. So this has to go 3 to 5 this way. This goes 3 to 5 this way. Okay. They have to be anti-parallel. They run the phosphate sugar background, and the purine and pyridine bases hold the DNA molecule together. They're the steps that hold the side rails together. Okay. So the DNA double helix, it's 2 strands of DNA form a double helix, a right-handed double helix, spins to the right. Bases are opposite strands, hydrogen bonds in together. Adenine always pairs with thymine, guanine always pairs with cytosine. The 2 strands are anti-parallel. One goes 5 to 3, the other has to go 3 to 5 prime. Okay. One turn, so one turn 360 degrees around is roughly 10 nucleotides. Then you make another turn, 10 nucleotides, then another turn as it spirals around. One complete turn accounts for 3.54 nanometers. Nano means 10 to the minus 9. So very small amount. And one nucleotide is about 0.34 nanometers in size. Okay. So those are the specifics for DNA. Okay. So this is what it looks like. This is the double helix. These are the strands. You can see every pair. Guanine pairs with cytosine. Adenine pairs with thymine. Cytosine. Oh, well, I drew the circle wrong. Back up. Adenine pairs with thymine. Guanine pairs with cytosine. Guanine pairs with cytosine. Adenine pairs with thymine. Okay. Notice adenine to thymine have 2 hydrogen bonds. Cytosine to guanine has 3 hydrogen bonds. Okay. The sugar phosphate background is alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, with the base extending from the sugar. So each sugar has a base extending from it. Okay. That's what extends into the center of the spiral. Okay. So 2 strands of DNA form a right-handed double helix. The bases are in opposite strands by the hydrogen bond. Okay. Adenine pairs with thymine, guanine pairs with cytosine. The 2 strands are anti-parallel. Okay. One strand is 5 to 3. So if this is the 3 prime end, this is a 5 prime end. If this would be the 5 prime end, this would be the 3 prime end. They run anti-parallel, just like here. This is your 3 prime end. This is your 5 prime. 3 prime, 5 prime. And these are anti-parallel to one another. They're opposite. Okay. And there are 10 nucleotides in every turn. So from this point to this point, for every turn, there's 10 nucleotides. Actually, it extends up. I'm sorry. Every turn starts, it wraps around up to here. So this is one turn from this point to this point. That's one turn. That's 10 nucleotides. Okay. This is what it looks like in a space filling model. Okay. You can see your background, and you see this pairing in between the sugar phosphate backbone, forming your bridges that hold everything together. Okay. This is chain number 1. That's chain number 2, with the bases in between holding it together. That's your sugar phosphate backbone. And the center is your hydrogen bonded base pairs in the core of the helix. Those are your purines and your pyrimidines. Again, adenine always pairs with thymine. Cytosine always pairs with guanine. Adenine to thymine is 2 hydrogen bonds. Cytosine to guanine is 3 hydrogen bonds. And the chains run anti-parallel to one another. One runs from the top down, 3 to 5. The other runs from the top down, 5 to 3. Prokaryotic chromosomes. Prokaryotic means, if you had biology, no nucleus. Like bacteria. Bacteria have a lot of bacteria have no nucleus. That's a prokaryotic cell. So prokaryotic chromosomes are DNA that are not confined to a nucleus, like a bacteria. Okay. So chromosomes are pieces of DNA. DNA contains the genetic instructions of an organism, what to make, what proteins to make. It contains every protein code that's going to be made. It determines the sequence of amino acids to build those proteins. Okay. So prokaryotes have a single chromosome. Okay, no nucleus, no true nucleus. Okay? Chromosome is circular DNA molecule that is supercoiled. So it's this coiled molecule. This double helix wraps around and folds on itself. So this is the cell, and inside the cell there's this circular DNA. No nucleus. It's coiled upon itself. So the helix coils back upon itself. There's about 40 sites with complex proteins attached, forming a series of loops inside that DNA structure. This structure is called a nucleoid. It's not a nucleus, it's a nucleoid. It's nucleus-like because it occurs in the cell. Okay? So you have these supercoiled DNA loops with its protein core around it, and all these DNA loops around each other. They coil around to form one long chromosome. Okay? Eukaryotic chromosomes. Eukaryotic cells, like human cells. We have a nucleus inside the cell, and we have multiple chromosomes. Okay? That's the difference between prokaryotic and eukaryotic. Eukaryotic, number and size of chromosomes vary. Like I said, humans are 46. The true nucleus enclosed by a nuclear membrane. We have a nucleosome. Okay, consists of strands of DNA wrapped around what's called histones. Histones are protein that we use to take this DNA when it spreads out and coil around these histones. We coil it around, it condenses it, and that's what forms a chromosome. That's why chromosomes look like little beads on a string. It's got all these little beads of what we call histones, these little proteins, and the DNA wraps around these histones, and that allows it to coil and form our condensed DNA. Now, when DNA is in the form of chromosomes, it can't be used. It has to open up and spread out so we can read that genetic material. It forms chromosomes for replication purposes. It can be divided into cells equally. So the string of beads is a larger structure called the 30 nanometer fiber. And with additional proteins, it becomes a 200 nanometer fiber. We add proteins to it. Okay? So this is your chromosome. This is what our chromosomes look like. Okay, they take these little proteins called histones and we start coiling around it. We coil, it gets tighter and tighter, and it condenses into what's called chromosomes. Okay, when it's spread out like this, that's when we're reading the code. We cannot read DNA code when it's condensed. There's no way to get at it. It has to unexpand itself so we can read that DNA code. Okay, all animals, plants, and fungi are eukaryotes. We have a nucleus. The number and size of chromosomes of eukaryotes vary from one species to the next. Okay, like I said, humans have 46 chromosomes, 23 pairs, 23 from the mother, 23 from the father. Okay, the adder's tongue fern has 631 pairs of chromosomes, 1,262 chromosomes. Okay, big difference. The closer the number of chromosomes are, the closer the species are on the evolutionary scale. Okay? But the chromosome structure is the same for all the organs that have been studied. So even though this has 1,262 chromosomes, it all has adenine to thymine, guanine to cytosine. They're all the same, whether it's in the adder tongue fern, or whether it's in a human being, or whether it's in the great apes in Africa, or the penguins in the Arctic. Okay, all the DNA is the same. It's all those same 4 bases: the purines and the pyrimides, same sugar phosphate backbones, same DNA linkage that holds it together. We use the same information to transmit genetic material between all species. Okay, just the number of chromosomes are going to differ and size are going to differ. So eukaryotic chromosomes, as you expect, are very complex. Okay, the first level of structure is what's called the nucleosome. Okay, consists of a strand of DNA wrapped around a small disc made up of histone proteins. So when we start to coil it up, we take these histones, it attaches to it, and we use that as a wrapping structure. Okay, this structure looks like beads along a string. You got the histones kind of situated along this line of DNA. The string of beads then coils in a larger structure called the 30 nanometer fiber. And then they further coil into a 200 nanometer fiber. And other proteins get involved in doing this. Then the full complexity of the eukaryotic chromosome are not yet understood, but there are probably many such levels of coil structures. We're not quite sure why we have different coil structures or what it really does. We just know it coils up for replication, then uncoils for use. Okay? So some human genetic disorders, when we talk about genetic disorder, it comes from DNA, are characterized by unusual chromosome numbers. Down syndrome. Okay? There's an extra copy of chromosome number 21. So if a patient has an extra copy of chromosome number 21, they have Down syndrome. The presence of this additional chromosome causes the traits associated with Down syndrome. Varying degree of mental retardation, some severe, some not so severe, flattened face, short stature. Okay? That's having extra copy of chromosome number 21. Okay? The presence of additional chromosome 18 causes Edwards syndrome. An extra chromosome 13 causes Patau syndrome. Both of these extremely rare result in extreme mental and physical defects and usually early death. That's why these people with this genetic don't live very long, that you don't hear about it too often. That's why Down syndrome, they still can have a long longevity of life. Okay. The presence of an extra copy of the sex hormone, X or Y, is not lethal. Okay. X and Y determine your sex. An XX is a female. An XY is a male. That's it. The female always donates an X chromosome in the egg. The male donates an X or Y. So 50% of the time a male donates an X, 50% of the time the male donates a Y. If the male donates an X, it's going to be a female. If the male donates a Y chromosome, it's going to be a male. Okay? Males with two X chromosomes and one Y X chromosome. So you have X, X, Y. Okay? Suffer from Klinefelter syndrome. Sexual immaturity and breasts start to develop. They have female sexual characteristics. That's Klinefelter syndrome, X, X, and Y. Males with an extra Y chromosome. Okay, X, Y, Y. Okay, are you unusually tall, as are women with three X chromosomes? Okay, so women with XY for males or XX for female. You're going to be extremely tall. Okay, women with only a single X chromosome is what's called Turner syndrome. That's only an X. Okay, include a short stature, like a web neck, and sexual immaturity, just like Klinefelter syndrome with the XXY. Okay. So there are different abnormalities that can occur because genetic isn't always transferred correctly. There are mutations that occur. Okay, so all other abnormalities in chromosomes, numbers are thought to be lethal to the fetus. Okay. So the X and Y's are the ones that are not lethal. The other ones are usually lethal. It's thought about 50% of all miscarriages are the results of abnormal chromosome numbers. That's a lot. So half of your miscarriages are thought to be caused by an ab-numbered normal of chromosomes. Okay? Only the X and Y's can get extra ones here, extra there, and not cause fatalities usually. Okay, the complete set of genetic information, okay, in all the chromosomes of the organ is called its genome. Genomes can be very complex. The karyotype is your division of your chromosomes. There are 23, 46 chromosomes in the human. There's 2 of every one, except there's 2 all the way to 22, and then 23 and 24 are your X and Y chromosomes. So this is your X and this is your Y. Okay. So the different chromosomes, all the chromosomes are called autosomes except for the sex chromosomes. Those are called sex chromosomes. So you have 22 pairs of autosomes and 1 pair of sex chromosomes. That make it for a total of 46. And again, that is only in humans. Okay. And that's what your chromosomes look like. Now we talked about DNA structure. Now we're going to talk about RNA structure. RNA structure is a little bit different. It uses a different sugar, has a different number of strings to it. Only has one length, not 2. And there's different functions for RNA. All right, has a sugar phosphate background. For ribonucleic acid is used, and it's linked by 3,5 phosphodiester bond. So similar DNA, it has a 3,5 orientation, has a sugar phosphate background, and instead of deoxyribophosphate for the sugar. It uses ribose for the sugar. Okay. Three main differences between RNA and DNA. Okay. RNA molecules are usually single-stranded. DNA is double-stranded. Ribose is the sugar instead of deoxyribose. And instead of thymine, you have uracil. Okay. So when you make, instead of having thymine, so the uracil is going to pair with the adenine instead of the thymine when it lines up. Okay. So RNA is single-stranded. It has ribose sugar instead of deoxyribose, and it has uracil instead of thymine as one of the bases. Okay. Base pairing occurs between uracil and adenine, guanine and cytosine. Okay. Hydrogen bond. Whoa. We got too far apart. Here we go. That's what I wanted. So the hydrogen bonding results in portions of the single strand that become double stranded. When RNA is formed, it's read as a single strand. In other words, RNA is going to form to your DNA, and that's going to form a double strand and then break off. We use RNA to read the DNA code. And instead of adenine pairs with thymine now, uracil is going to pair with adenine. And guanine and cytosine still pair together. Okay? So these are three big differences right here. Single-stranded, ribose sugar instead of deoxyribose, and uracil replaces thymine. All right. So fooling the AIDS virus, the look-alike nucleotides. Okay, remember how viruses work? Coronavirus, the AIDS virus, they all work the same. They're not alive. They get injected in the body, you have to use the cell's metabolism to make it. They hijack the cell's DNA and trick it into making more of the virus. So the virus that is responsible for the acquired immune deficiency syndrome, AIDS, is the human immunodeficiency virus, HIV. That's the virus. HIV is a member of the family of viruses called retroviruses. Okay? They all have single-strand RNA as their genetic material. So inside the virus, so if this is your virus capsule here. Okay? Inside the virus, there's a single strand of RNA. That's the genetic material. That's it. That's what's inside the They have a polyprotein coat around it. And the little attachments on the outside, they can travel, and it has a single strand of DNA inside of it. Okay? The RNA is copied by a viral enzyme called reverse transcriptase into a double-stranded DNA molecule. So the RNA in the cell is injected into our cells, and the human cells copy this viral enzyme using reverse transcriptase to form double-stranded DNA. And now we got the viral code in us. Okay? This process is the opposite of what usually happens, which states the flow of material is from DNA to RNA. Normally, the DNA goes to RNA, goes from messenger RNA to transfer RNA, and then we make proteins with that. Okay? So we're flowing from DNA as our code going to RNA. It's a retrovirus because it goes backwards. Retrovirus is backwards virus. It takes its RNA and it uses an enzyme called reverse transcriptase, and it forms a double-stranded DNA from RNA. It goes the opposite direction. Both these viruses reverse the flow, so we're going from RNA now to DNA. These viruses are called retroviruses, which means backwards virus. So the process of producing DNA copy of the RNA is called reverse transcription. Transcription is the act of copying DNA to RNA. We transcribe it. Okay, so it's called transcription. So reverse transcription is copying the RNA to go into DNA. So reverse transcriptase is the enzyme that allows that to happen. Okay, so because our genetic material is DNA, and it's expressed by the classical DNA to RNA to protein, our cells have no need for reverse transcriptase. We don't have that enzyme. We don't need it. Why do we need to go backwards and make DNA from RNA? We don't. We just go from DNA to RNA to our protein. Okay, so HIV reverse transcriptase is a good target for antiviral chemotherapy. Again, you want to target something that is not in human cells. You want to target something that is specific for the virus, right? You want to kill the infected cell. You want to kill the virus without killing the patient. That's your net goal. Okay, so we know that reverse transcriptase, we can target that because that is not part of a human cell. If we can kill off reverse transcriptase, we're not going to hurt human cells because we don't use it. We don't go from RNA to DNA. Okay, so if we can kill that, the virus uses reverse transcriptase to take its RNA to go to DNA. So if we stop it at this point, if we have something that can kill or eliminate reverse transcriptase, we can stop the virus from replicating, in theory. Okay? I mean, everything here is in theory. It's not, it doesn't actually work 100% like that, but we have some good drugs for that. So inhibition, reverse transcription should kill the virus, but have no effect on human host. That's not completely true. It does have some effect. There are side effects or other things that happen, but in theory, this is great. That's what you want to do. You want to target something that is specific to the virus and not specific to human cells. Okay? Many drugs have been tested to selectively inhibit HIV reverse transcriptase. That's what we're going after. If we can inhibit that, we can do good. Among these is a DNA chain terminator 3-azido 2'3' dideoxythymidine, commonly called AZT. AZT was one of the first drugs we used in treatment of AIDS. And AZT was extremely expensive drug back then, and it goes after this reverse transcriptase. Where are we at? Yeah, it goes here and it inhibits reverse transcriptase. So does not affect human cells because humans don't have reverse transcriptase. It affects the viral cells. Okay, so how does AZT work? It's one of the many drugs that looks like one of our normal nucleosides. These are called nucleoside analogs. Analogs are something that have the same structure and are very similar. So a nucleoside is just a nucleotide without any phosphate groups. So our nucleoside is the same as a nucleotide. A nucleotide, remember, has a sugar, phosphate, and a base, nitrogenous base. Okay, so a nucleoside is just our sugar and our base. There's no phosphate groups attached to it. The analog is phosphorylated by the cell and then tricks the polymerase, into this case viral reverse transcriptase, into incorporating into the growing DNA chain. So it looks like or mimics one of the nucleotides. It gets put in the chain as it's going from RNA to DNA using reverse transcriptase. It sticks this in the chain in place of the normal nucleotide and makes it non-functional. Okay, so AZT is a nucleoside analog that looks like the nucleoside thymidine, except in the 3' position of the deoxyribose sugar, there's an azide group and 3 groups rather than an OH group. And that's the difference. This is thymidine. This is a normal nucleotide that gets incorporated into the DNA that's being made by reverse transcriptase. Okay, this is thymine. This is your ribose sugar, or your deoxy ribose sugar. And the only difference, if you look carefully at it, the only difference, this has an OH group here, this has an azide group here. Azide is N3. Okay? So it's incorporated in it. If this gets incorporated in the DNA becomes non-functional because this doesn't form the bonding necessary to make the chain. So it disrupts the transcription process. The DNA is faulty for making the virus now because we incorporated a bad nucleotide into the sequence. Okay. So the 3' OH group is necessary for further DNA polymerization. Remember, we add at the 3' group. We have 3', 5', 3', 5'. We go to the 5' group, we add our next sugar, our sugar phosphate background. It's always 3' to 5', 3' to 5', 3' to 5' as this expands. Okay, and this adds at that OH group in the number 3 position. So now instead of the OH group, we have an N3 group in that position. We can't add to it. So the chain stops. So it doesn't make the DNA. It puts the fake nucleotide in there. And when it goes to add the next one, it can't do it. The sugar phosphate backbone can't proceed because now instead of having our OH group where we can do a dehydration reaction, remove water and get our linkage, now we have an N3 group attached. There's no way to get water out of N3. You can't dehydrate that bond. because there's no oxygen there, no hydrogen there. Okay? So you can't get rid of it. So it stops the DNA from there, stops the DNA from replicating, stops the DNA from replicating. The virus can't make its own DNA and it can't replicate itself, and it dies. So the viral RNA cannot be reverse transcribed into DNA. The virus will not be able to replicate because it can't use the DNA and is considered dead. That's how AZT works, in theory. Okay, so this is your chain termination. So in other, you're adding oxygen and your phosphate group and your base, and you got your OH group. So you're putting that into the phosphate group, then to your sugar. Okay, so the sugar and phosphate, you form your sugar, phosphate background. That's your backbone. Okay, where there was an OH there before, now we have an azide group, which is N3. So no OH. You can't dehydration reaction, you can't form the phosphodiester bond. Okay, and then the chain cannot continue. So you get chain termination. Okay. That's how it works in a nutshell. So AZT is particularly effective because the HIV reverse transcriptase actually prefers it over the normal nucleotide thymidine. AZT is not a cure if you're infected with a virus. At best, it prolongs the life of the person with AIDS for a year or two. Okay, that's AZT by itself. We have other treatments available now where we mix it. Eventually, AZT has a negative effect on your body. Okay, it doesn't work exactly as we just said. Okay, the cells of our bone marrow are constantly dividing to produce new blood cells, red blood cells to carry oxygen to tissues, white blood cells for our immune system, and platelets for blood clotting. For cells to divide, they have to replicate their DNA. So the DNA polymerases, which allow us to replicate our DNA, okay, also accidentally incorporate AZT into the growing DNA chains. So, okay, even though we're using, it's attacking the reverse transcriptase, we also have an affinity for it, okay, for our own DNA chains. So then we incorporate that in our own DNA chains, we can't replicate our own DNA chains. Okay, because it still puts that azide group at the end, so we can't continue to replicate our own DNA. Okay, so the bone marrows begin to die. Bone marrow begins to die, we have serious problems because our bone marrow is our red blood cells, so we get anemia. Our bone marrow is our white blood cells. We get our autoimmune system starts to work or stops working. We get autoimmune disease, right? And the platelets start to be decreased, so we bleed out. We don't have platelets to clot our blood. So there's some serious problems with AZT. It's not all that it's cracked up to be. This can result in anemia and further depression of our immune response because of the red blood cells and white blood cells and platelets were all produced in the bone marrow, which we cannot replicate in bone marrow anymore because AZT is attacking it. Okay. Another problem arisen with prolonged use of AZT. AZT-resistant mutants of the virus appears. So with any other, like any bacteria or any virus, it's going to evolve. It's going to change depending upon the conditions it's under. Okay? It's well known that HIV is a virus that mutates rapidly. Okay? Very fast generations. Some of these mutant forms of virus have an altered reverse transcriptase that will no longer use AZT. And those are the ones that are going to be selected by use of AZT. We're going to select those out, and that's going to be the common form now, is the one that has altered reverse transcriptase. When these mutants appear, AZT is no longer useful in treating the infection because it's not going to be incorporated anymore into the growing nucleotide chain for DNA. Fortunately, research with other nucleoside analogs, alternative types of antiviral treatment, and combination of drugs have proven more effective means of treating HIV infection. HIV, we still use AZT today. We use it in combination with other drugs, though. We do not use it by itself. In 1987, that was 30 years ago, 35 years ago, AZT was costing the patients $10,000 a year. At that time, that was the highest priced drug in history. I'm sure it's been passed by other ones. I don't know what the new high one is. All right. Any questions with AZT treatment or the analogs or the nucleotides, sugar phosphate background with the nucleotides that connect? All right. Normal DNA replication. DNA must be replicated before every cell divides. Because if you take one cell to two cells, you've got to have 46 chromosomes in each. So you've got to duplicate the DNA and split it between the two new cells. Okay? Each daughter cell has to inherit a copy of each gene. Cells missing a critical gene are going to die, because that's what determines all our proteins that function. So essentially, the process of DNA replication produces an absolutely accurate copy of the original genetic material. That's what's supposed to happen, but that doesn't always happen. Things can cause mutations. UV radiation, sunlight causes mutation. It causes skin cancer. We cause mutations in their DNA. Certain kinds of chemicals cause alterations in our DNA. Okay? Certain drugs. Okay? Mistakes made in critical genes can result in lethal mutations. Some mutations are not lethal. Some mutations are beneficial. That's how evolution occurs. Okay? All right, so the structure of DNA molecules is just the mechanism for accurate replication. An enzyme could read the nitrogenous bases on one strand of a DNA molecule and then add complementary bases to a new strand. So if DNA, which is double-stranded, you got the double strand, if this opens up these strands and then you got this area exposed to these areas, another strand can come here and make a complementary copy. And it can form a new DNA strand. And that's what happens. When it opens up, we use one of the strands as a base plate, basically, and then we make a complementary copy. And that becomes a new DNA strand. Okay, a newly synthesized DNA strand by using complementary bases. Product of this strategy would be a new DNA molecule, okay, in which one strand is the original and the other strand is the daughter strand. So half this would be the original here, and this would be the daughter strand being formed by adding complementary bases to it. Okay. This is called semi-conservative replication, because one half of it's staying the same and the other half is being synthesized by a complementary strand. Okay. So this is a better picture here. You can see you have your two windings around. Okay. Then you have your original and you have your daughter strand. That opened up. This is the original and this is the daughter strand. Okay. And again, then it duplicates again and again and further on. And everything just gets copied and you make one strand stays the same. One strand is the complementary strand. And by doing that, you make copies of your DNA. Okay. So here's your DNA double helix and it starts to open up. Okay. You can see as it opens up, this is your replication fork, the fork in the road. And it's replicated. You can see where your adenine was paired to thymine, cytosine was paired with guanine, adenine to thymine, guanine to cytosine. And now hydrogen bond has been broken and those open up. As they open up, a new strand complementary can form. So this blue strand here is being added to it. So if you have a guanine sticking out, you put in a cytosine. An adenine sticking out, you put in a thymine. And this makes an exact duplicate, two exact duplicates of the entire DNA structure. Okay, you can see how that's going to form. Okay, so it opens up the replication fork with certain enzymes, allows the complementary strand to form inside to the other one, and it's going to form two copies of DNA, and it can split off one into each cell. So you get double the number of chromosomes, and they're going to split apart. Half go to one cell, half goes to the other cell. Okay, so you can see the purple is the original template, the original strand. The blue is the newly synthesized strand on both sides. That's why it's called semi-conservative. One strand is staying the same, the other strand is being synthesized by using complementary nucleotides. Okay, so the product of DNA replication is going to be two new DNA helices. And they're going to, so you start with a one strand here. And again, it's anti-parallel. One goes 5 to 3, one goes 3 to 5. These open up. The purple is your original. The blue is the newly synthesized complementary strand. And you can see those are two exact duplicates of your original. Okay? And they're both anti-parallel, 5 to 3 and 5 to 3. So each helix now has two DNA strands that once are formed. One strand is from the parental DNA, which is purple. The other blue is the newly synthesized DNA strand. An exact duplicates. And that's how DNA is duplicated. Bacterial chromosomes is a circular molecule of DNA. Remember, circular molecule because it doesn't have a nucleus. It is a prokaryote. Okay. So it is a circular molecule that's kind of folded upon itself in loops. All right, there's approximately 3 million nucleotides. Okay, so DNA replication begins at a unique sequence, which is your replication origin. That's where replication starts for a prokaryote. Replication moves bidirectionally, 500 nucleotides per second. So in other words, it starts, if you have your circle here, it starts here, it moves both directions. Okay, that cuts the time in half of what it would take to duplicate it. It allows you to be much more efficient, much faster. And once the strand opens up, it puts in a nucleotide, 500 nucleotides every second. That's really quick. That's amazingly fast to keep track of. The position where the new links are added to the growing daughter strand is what's called the replication fork. That's the division point. That's where the DNA splits into two strands. Where you have your original strands and your daughter strand starts to form on it. And again, it's going to be bidirectional. It's going to form both directions across the circular DNA. And it's going to 500 nucleotides per second in both directions. As DNA synthesis moves bidirectionally, there are two replication forks. One moving one way, one moving the other way, right? They're moving two different directions here. So you have two replication forks where this opens up and allows the strands to duplicate. Okay. So this is your original. Okay. This is your origin of replication. That's where replication is going to start. So this starts to open up and it proceeds this direction and this direction. Okay. When it gets done, you have this connection here where everything's been split. It's gone both ways. It's completed the circle and then it divides. And now you have two forms of bacterial DNA that are exactly the same. One from was not been modified. The other is the one that has been synthesized from the complementary strand. And again, it has the same basis that we have in human DNA. Adenine to thymine, cytosine to guanine. Okay. That's how bacterial DNA replicate. And again, it's 500 nucleotides per second. I mean, so you're basically covering 1,000 nucleotides in a second, 500 each direction. That's going pretty darn quick. Okay. The first steps in DNA replication. Okay. The first step has to be separation of the strands. If the strands don't separate, you can't duplicate them, obviously. So the first step has to be separation. And this is accomplished by a helicase. Remember, ASE means an enzyme. Okay. The helicase breaks apart the hydrogen bonds between the base pairs. Remember what's holding those DNA strands together. Adenine's bonding to tymosine with 2 hydrogen bonds. Cytosine's binding with guanine with 3 hydrogen bonds. Helicase breaks the hydrogen bonds between the base pairs. And that's what holds it together. And that's what allows it to open up. So that's your first step, the helicase enzyme. Positive supercoiling results when hydrogen bonds are broken. This is relieved by topoisomerase. Topoisomerase. Okay, it's broken up by that enzyme. Okay, so you get supercoiling results when the hydrogen bonds break. This tends to coil together, but this topoisomerase tends to break that apart to keep the strands open for the complementary strand. When supercoiling is relieved, the single strand binding protein, protein binds to each separate strand to keep them apart. Okay, so what's happening is you have your DNA and it's paired, going around. Okay. Helicase breaks those bonds that are holding this together. The hydrogen bonds get broken between there. Once those hydrogen bonds get broken, this is going to tend to supercoil around itself. Okay. Another enzyme, topoisomerase, comes in here and it stops this from supercoiling around itself and leaves it open. Once it's open, the single-stranded binding protein, what it's called, binds to each separate strand, and it keeps each separate strand from supercoiling again and keeps it open. Once it's open, now you can synthesize a complementary strand. And primase, okay, catalyzes the synthesis of 10 to 12 base pieces of RNA to prime the DNA replication. You got to get it started, just like you prime a gas motor. Okay, you put gas in it, get it to run. You've got to prime your DNA to get it started. Okay, so primase is an enzyme, and you get like a 10 to 12 base piece. So 10 to 12 bases get copied, and this kicks it off. This gets the DNA replication to start. Okay, so you've gone through 3 enzymes already. You've gone through helicase, which breaks the hydrogen bonds between the nucleotides. Topoisomerase, which causes the supercoiling when it breaks apart to become released and open back up again. So once the helicase breaks it apart, it tends to coil upon itself real tightly. The topoisomerase tends to break that up and let that expand back open. And then the primase gets you a 10 to 12 sequence group to start with the nucleotides and gets the replication started, gets it primed. So those are your 3 enzymes. And the other protein involved is a single strand binding protein, which binds to the strands that it can make your complementary set by helping to keep them open. Okay. So this is what we got. Here's your replication fork. This is where you got your division starts. This is a 5' end, 3' end. Okay. So we have the replication fork. Top up on, where's our first one? First one is helicase. Helicase breaks the hydrogen bonds between the nucleotides, between the bases. Okay. And then topoisomerase, okay, stops the single strand that's been released from supercoiling. Okay. And where's our, we don't have our protein here. Oh, here it is. And then once it starts from coiling, the single strand binding proteins bind to the strand and keep it open and prevent the supercoiling. Okay. Then the primase kicks in, okay, and it starts by dumping a 10 to 12 section and coating that, and that kicks off our replication. Okay. So all these blue ones that we just circled here, we just talked about, those are all involved in the first step of DNA replication that we just talked about. All right. That's the first step. Now we go to the next step. Okay. After the first step is completed, DNA polymerase 3, okay, reads the parent strand, okay, the template, catalyzes the polymerization of a complementary daughter strand. So DNA polymerase 3 kicks in, and this thing starts to read the whole strand and put that complementary strand all back together. Okay, so in the polymerization reaction, a pyrophosphate group is released as a phosphoester bond is formed between the 5-phosphor group of the nucleotide being added and the previous 3' OH of the nucleotide in the newly synthesized daughter strand. All that means is it's putting the complementary base onto the DNA in the anti-parallel direction. So it's going to run from 3 to 5 if the strand is running from 5 to 3. Okay? And it's just going to add those right to that complementary strand. So based on the bond formed in the polymerization, this is referred to as 5-3 synthesis. Starts at the carbon 5' and bonds to the carbon 3' of the sugar group. And that way you get your sugar phosphate backbone of the newly synthesized strand. Okay. So this is a DNA polymerase reaction. This is your 5' end. So if this is the main strand, okay, and this is going to be the copy. You have cysteine, so it's going to pair with guanine. You can have your base, and it's going to be a 3 5 connection. Okay, because this is your number 1. That's number 2, 3, 4, and 5. When we do that. So you're running a 3-5 connect, 3' to 5'. The exact opposite of the parent strand. This starts at the 5' end, it starts at the 3' end. They run anti-parallel. Okay, and as they form, they bind up. The adenine pairs with thymine, guanine pairs with cytosine, okay, and they add their base and their phosphate groups. Okay? And you form an anti-parallel chain with the new exact duplicate of the original one. Okay. So factors that influence DNA replication. The two DNA strands being replicated are anti-parallel. Just like we said, the DNA polymerase 3 can only catalyze in the 5-3 direction. Okay, so the original is in the 3-5 direction. Okay. The replication fork moves in one direction while both strands replicate simultaneously. Okay. So the DNA fork opens it up, moves in one direction, splitting it open, and then both those new strands are being replicated at the same time. Okay. Small RNA primers, okay, are needed for a starting point of DNA replication. So you gotta get something to kickstart it. So there's 2 different mechanisms for replication of the 2 strands. The leading strand can be replicated continuously. The opposite strand, was the lagging strand, is replicated in segments, okay, or discontinuously. Okay? So it can either replicate continuously as strands open up right down the line, or it can open up and they can replicate each section and then spin them all together. Then kind of basically attach them all together. So 2 different ways that can happen. Okay? So a single RNA primer is produced at the replication origin. Okay? The part where replication starts. DNA polymerase 3 continuously catalyzes the addition of nucleotides in a 5-3 direction. That DNA polymerase continually adds to it. So many RNA primers that we use are produced as replication forks, move along the molecule. They move along at different points will start, and replication begin in different areas that way. So DNA polymerase 3 catalyzes the elongation of the new strand, again, in the 5-3 direction. As the new strand encounters a previously synthesized new piece, synthesis stops at that site, and those pieces are joined together. So if you, oops, what did I hit? Okay. So in other words, if the replication fork starts at one point, then another point. You get those RNA pieces that start the DNA to kick off, the primers. Okay. Once it goes to the point and reaches the original, the next one, it stops replication there and then joins those together. So now you have a continuous strand of new DNA. Okay. So that's where it stops. The process repeats with another primer at a new location of the replication fork until it meets the next one. Now you got another section and it joins it together. So the final step is to remove the RNA primers, and we use that with DNA polymerase 1. Okay? And then fill in the gaps where the RNA primers were. And we use DNA polymerase 1 to do that also. Okay, the sealing the fragments into intact strand of DNA is DNA ligase. So all those strands that we produce now with the primers that kicked them off, we kick those primers off, we got strands, we fill the gaps in with DNA polymerase, and then DNA ligase seals it fragments into an intact strand of DNA. So it's completely intact. Okay. That's how it works. You can see this is the detailed view of it. This is your leading strand. Again, 3 to 5 directions, they go in opposite directions. This is the lagging strand. You can see how this strand is going here, this strand is going here. When it meets it, it stops, because here's your DNA primer that kicked it off. Okay. Once it meets the other chain, it stops, kicks off the DNA primer. The primase fills in the gap, and then the ligase seals it all up and becomes one DNA strand. Okay, so this is the RNA primer down here, and this is your single-stranded DNA binding proteins that keeps those bands open so they don't supercoil on you, and you can still keep adding things to it. Okay, but you see your DNA polymerase 3 is adding nucleotides to those open strands. And it occurs at the replication forks, and they could have several replication forks because the RNA primer here can kick off and start the DNA to synthesize. And once it meets at the other chain, it stops. The RNA primer is kicked off. The DNA polymerase comes in and replaces the RNA primer with DNA nucleotides. And the ligase puts everything together. And now you got a complete copy of your DNA. Okay. So DNA, the DNA polymerase 3 reads discontinuously because they have multiple replication forks, and it reads in the opposite direction. Okay. It allows you to go from that anti-parallel synthesis of the new chain. Let's do, almost got it here. Let's see, because it's critical to produce an accurate copy, okay, it's very important to avoid errors in the replication process. So in addition to catalyzing replication of DNA, DNA polymerase 3 is also able to proofread the newly synthesized strand. It goes through and just makes sure everything is correctly translated by the complementary strand. If the wrong nucleotide has been added to the growing group, I mean, you're replacing 500 nucleotides a second, you can make a mistake. Okay? So if the wrong nucleotide has been added to a growing DNA strand, it is removed and replaced with the correct one. Okay? And that's one of the functions of DNA polymerase 3. It goes through when you're done, reads the strand to make sure the complementary strand is correct. If one's wrong, kicks it off and replaces it. Okay? That way you have a very accurate copy of your parental DNA, your original DNA.